Hello everyone, welcome back to the cell signaling course, and we'll continue from where we were in the last class, where we discussed different modes of receptor tyrosine kinase signaling which are dependent on phosphoinositides. We discussed the function of PI3 kinases, how they help generate PIP3 entities which takes the signaling in a given direction. We also discussed how the PLC gamma activity, which will break down PIP2 into DAG and IP3, will take signaling in different direction. And in some sense we discussed that, in some sense we appreciated that the PI3 kinase signaling and PLC gamma signaling are working in opposite directions. Today we will discuss aspects of contact-mediated bidirectional signaling, which is shown by a special class of receptor tyrosine kinases. So we all understand by now that receptor-mediated signaling, including receptor tyrosine kinase, it plays very critical role in growth, development of organism, survival, and if something goes wrong with them, lot of disorders which are there, right? We also understand that all these things which we are discussing until now, the ligand was probably synthesized at a far off distance, right? It was more like a long distance signaling. But when it comes to contact mediated signaling, we are talking about short range signaling or juxtaposed signaling, where two cells which are in the vicinity, they are signaling to each other. That is where it sometimes becomes very important in contact mediated signaling that the signaling is bidirectional in nature. We will see in today's class that how some of these signaling which is present in the system will be helping in the tissue organization, early developmental patterning processes, boundary formation and exon guidance, which probably we did not appreciate until now. So what is the need of this contact mediated signaling? I briefly told. If you think about a cell which is growing in the vicinity of other cells, depending on which stage we are talking about of development, there will be signaling in both direction. You need signaling in both directions so that the cells which are in the periphery, they know how to develop and what to become, and the cell in center, this probably is a master signaling cell, telling all neighboring cells not to become what this master signaling cell wants to become. If I take for example, suppose the cell in center wants to become a neuron, and it does not want neighboring cells to become the neuron, so it will give signal accordingly, so that neuronal signaling or neuronal fate is inhibited. Or for that matter, think about a cell which is becoming secretory type. The center cell is becoming secretory type. It does not want other cells to become secretory type, so signaling will be going in such a manner that, okay, I am becoming the secretory cell, you don't become a secretory cell, you can become a support cell and so on. So here the contact between the two cells is very important. If you recollect when we were discussing the root hair development in plants, and we took example of receptor like kinases over there. So one cell which was in the outer side, which was in contact with two cells versus one which was in contact with a single cell. So the one which was in contact with two cells, that became the root hair cell, and the other one became non-root hair cell. So this contact over there, at least it was not physical contact between the two cells, rather secretory peptides were coming from this cortical cell, deciding which cell becomes the non-root hair cell and which cell becomes the root hair cell. But somewhere there is a component of how many contacts it is making was there, right? Here we will see in case of contact mediated signaling, which is mediated by or which is activated by the receptor tyrosine kinases of a very different type. So the receptor here is called EPH because of its understanding the way it was described in the first time. It is erythropoietin producing hepatocellular cell. So the receptors are present in these cells and it's called EPH. Then you have the ligand for this, which is called EPH receptor interacting or Ephrin, which is. Now if you look at their combination in which they come together, and we simply make a small block of how these receptors and ligands are. So there are two types of receptors, type A and type B. Similarly, there are two types of ligands, type A and type B, right? And there are multiple of these genes which are present in animal kingdom which code for EPH receptors and Ephrin-like molecule. And there is a crosstalk which exists over there, EPH, if I say EPH A, EPH B, Ephrin A and Ephrin B. So there is a good amount of crosstalk between this two. This is generally a normal interaction which you would see. Then there will be crosstalk of this and this. There is an interaction of this type. So multiple of these interactions are there. So again the permutation and combination comes into picture. So the two cells which are in the vicinity, they are expressing both the receptors as well as Ephrin. It makes the signaling even more complex. You will be seeing in coming slides that how it is very different from the other cells. So let's have a look at the EPH Ephrin signaling, which is a bidirectional, and take a scenario of a cell junction. So the cell junction here, the space, this is where you have the, suppose I say EPH receptor, they have extracellular domain. When we were discussing multiple different types of extracellular domain in receptor tyrosine kinases, one of the domain was fibronectin type 3. So this kind of domain is present in the extracellular domain over here. Then you can imagine that if suppose I say EPHA, it does not mean that it does not express EPHB, it will also express EPHB. Or if it is EPHA, it will also express Ephrin of cognate type, and it engages in interaction in lateral way as well as the sideways. If I say Ephrin and this is your EPH, right? The difference between Ephrin A and B is that A is much small and has tethering into the plasma membrane. It lacks any significant portion inside the cytosol, no cytosolic region. So it is dependent on another co-receptor. Sometimes there is an example of P75 family of receptors which work along with Ephrin A and gets into complex with the Ephrin EPH receptors. On the other hand, the Ephrin B type receptor, if I take example of Ephrin B type receptor, they have intracellular domain and where they can be phosphorylated on tyrosine residue or serine residue. So, when there is a signaling going on between the two cell types, you would see that the kinase domain which is present in the EPH receptor will phosphorylate itself, functional engagement will phosphorylate the cytoplasmic tail of the type B receptor, and signaling will go in both directions. There is something called forward signaling and there is something called inverse or reverse signaling. So, here, and I told this long time back, the receptor bearing cell is actually doing the signaling. That was the case when the ligand was soluble and ligand was not membrane bound and also not having any co-receptor kind of situation. So, if you take EPH Ephrin signaling into account, EPH bearing cell, the one which is receptor type, will show you forward signaling. And the one which is going into the Ephrin, that is your reverse signaling. There are multiple molecules, as I say, the serine phosphorylation happens, tyrosine phosphorylation happens. So, cells will have the soluble tyrosine kinase activity, soluble serine threonine kinase activity. They will end up phosphorylating the cytoplasmic tail of the Ephrin B type receptors. Okay. Now, what happens, how this engagement is lost? So, this extracellular domain which we see here, if I can just arrange it here. So, extracellular region of the receptor, they have a sequence which can be digested by the help of a protease. So, protease dependent cut which happens thus disengages the two cells which are in forward revert type bidirectional signaling. Let us see further aspects of this signaling where, as I told you, you take a scenario where one cell is here, other cell is here, and this cell is actually expressing the receptor. I also told that both cells can actually express the receptor, and this side is expressing the Ephrin B type ligand, and this is expressing type A ligand. So, the cross phosphorylation event which happens, they activate the signaling, and once the signaling has to stop, the digestion will happen here by a protease. This is push and pull, push and pull. The total force which is generated by the two cells will eventually decide how the signaling is terminating. And in this process of push and pull, you will see repulsion happens. Sometimes the signaling starts and cell realize that this is not the right place to be. So, it actually comes out because of the pull force it will come out, and for that you will realize that cutting at this end to disengage the two cells is going to be very important. In case this cutting cannot happen, maybe for whatever reason, whether the complex is too big or it is actually undergoing some sort of lack of these enzymes which cut there, then there is endocytic event which is activated. And as I said that here you notice that the receptor and ligand are from two different cells or neighboring cells are engaging in signaling. So, this is your productive signaling. And often this productive signaling tells two neighboring cells that they need not be engaging in interaction, and that is where repulsion happens, and that is where productive signaling leads to repulsion. What can also happen in EPH-Ephrin signaling is that receptor is here, if I say EPHR, and there is ligand on the other surface, Ephrin, and if same Ephrin is present here in the cis cell, this is trans cell, this is cis cell, so it will go into productive engagement and signaling. So what will happen in this case, the signaling will not go across the cell, rather it will remain in the same cell, right. So presence of cognate ligand in the vicinity of the EPH receptor and engaging in this kind of EPH-Ephrin binding on the same cell leads to inhibition of signaling, and this is called cis inhibition. So it will not get into the signaling further. Like I said, productive signaling will lead to the repulsion. So if cis inhibition happens, productive signaling will not be there. One more thing which is very commonly seen with EPH-Ephrin signaling is that they undergo a lot of oligomerization type of event to build a big clustering signaling clusters. So what can happen over there is that the cis interaction which is there as well as the oligomerization will lead to this something called the clustering thing which I told you. So you look at the situation where you have the cell which has the receptor. If I now make a oligomer of these receptors, you will also have in between the, you remember the dimerization was one of the key event towards the activation of the receptor. Here oligomerization will lead to super activation of the entire thing. So this is a complex which is built up over here with Ephrin and EPH like molecule. So it will start with one set of receptor and ligand, but slowly it will grow into these multimeric oligomeric complexes. Now I can think about the force which is being generated over here. So obviously the one which makes a bigger cluster will end up generating more force. If for some reason this digestion is not happening, then the one which has more set of receptor or ligand and able to generate more force will do the pull force, more of a pulling, and thus culminating the signal. That will be very important for such signaling events to go on. So to understand this more clearly, we saw, if I say monomer receptor, it is engaged with monomeric ligand. From here you can have dimer, here then multimer and oligomer structure, and then this cell with the EPH is pulling so that you can have forward signaling and termination, or it can be the reverse signaling in Ephrin expressing cells. So what you understand that basically here these clusters are signaling and lot of movement which is happening inside the cell. One of the domains which is found in the cytosolic region is called PDZ domain. This engages in multiple protein-protein interaction. And what is the outcome of these protein-protein interaction? Things moving together, things going away from each other, things are brought together, and this is where the movement of cells leads to the patterning of tissue. Similar type of receptor expressing cells come on one side and the ligand expressing cells move to the other side because of this repulsive signaling. Now, what exactly happens in the repulsive signaling? As I said, here take a scenario where you have cells. This is expressing Ephrin, and you have the other cell which is expressing EPH. Once they get into this productive signaling, you make a, suppose you make a bigger cluster, these engagements happen, and as I said, if the cleavage is favored, suppose ligand cleavage was done, so what will happen? That this tail will remain here and this entire complex of Ephrin extracellular tail and the entire EPH will be taken inside the cell. What will be the resultant situation? That you will have the membrane and eventually you will have the endocytic vesicle, which will have the ligand and this receptor together. So, here endocytosis is happening, and that is ensuring that this productive signaling has met its end. The two cells, which is ligand bearing or the receptor bearing, they are now separated because what was holding them together has been resolved. In one of the cells, it has moved. The other situation is where the something called transcytosis which may happen. So, what will happen in transcytosis? Consider the same scenario of the two cell membranes, multiple of these ephrins which are over here, and right, they engage in productive signaling, and here for whatever reason you are not having that protease activity. Remember, any protease activity is not good. So, this molecule which is present and outside, that will not be free to cut receptor before it can be engaged into the signaling. So, some way this enzyme has to be kept inactive, right? And in situation which we are discussing here, this inactive enzyme is not activated. So, it is not going to resolve by digesting the extracellular domain of the ligand. Rather, what will happen? This is where push and pull will happen. Now, think a scenario where the push-pull happens and a bigger cluster is formed here, and you have Ephrin B type receptor which engages more molecule internally, which are tethering things to the cytoskeleton, and it generates excess pull force. So, what will happen here? That this cell will now take the entire complex with the entire receptor with it from the other side. So, basically you will have transendocytosis. Otherwise, if this cell ends up winning, then it will have the vesicle which has this scenario where both molecules are coming inside. So, this is also example of transcytosis. So, entire cluster of the receptor is taken back into one of these cells, and thus it will culminate the signaling, because the productive engagement is not possible at the cell surface, because ligand and receptor both have been taken into one of these two signaling cells. The removal of the receptor in general is one of the regulatory way to ensure that signaling is culminating after it has been activated at the cell surface. So, what we have understood in this with this unique type of receptor tyrosine kinase, that EPH is the receptor which is type A and type B, Ephrin is the ligand which is type A and type B, and type A is much smaller, it does not have a cytoplasmic tail, whereas B has a big cytoplasmic tail, and they often engage in repulsive signaling, basically, which means that once they get into the productive signaling, the changes which happen into both cells, which are part of the bidirectional signaling, they now are removed from this signaling cluster. EPH Ephrin, it is presenting a unique example of bidirectional signaling in case of receptor tyrosine kinase signaling. The complex of the receptor and the ligand, they form oligomer. They also show cis inhibition because the presence of same ligand on the same cell surface, and culmination of signaling by this transendocytosis process. Further, here you will see that cytoskeletal rearrangement is required so that cells can go back not to going into the productive engagement. They move away, they remain away from the contact zone, and so on. For that, cytoskeletal rearrangement is very important, and many of the PDC containing domain facilitate this protein-protein interaction, which is part of the signaling in EPH Ephrin signaling. Thank you.